So good morning, good afternoon, everybody. It is our great pleasure to start again the cycle of seminars of physics of the physics department of Ecole Polytechnique after almost 1 year and a half of interruption. So I'm glad to I'm glad to see you again for this first seminar of this year. We are very pleased to welcome Valentina Emiliani, who comes from Institut de la Vision in Paris. Valentina, as you will hear in a few seconds, comes from Italy, but it was a long time ago. You obtained your PhD there in the 90s. And after that, you went to Berlin, and you were first involved in quantum physics. In 2002, you joined the Jacques Monod Institute in Paris, and then there was a switch, and you worked as the interface between optics and biology. And ever since, you're still working on this, at this interface between two different kind of sciences, but which is very rich, as you will show in a few minutes. So you joined the CNRS in 2005, and after that you worked during 15 years or so at Université Paris Descartes.
Yes. And
in 2018 or 19, you joined the Institut de la Vision in Paris, where you are now head of the photonics department. Your domain of work, of research, is the wavefront sensing applied to biology. So we'll let you describe in full detail your work, of course. But I would like to say simply that your work and your research has allowed you to be granted a certain number of awards, among which I would like to mention at least the silver medal from the CNRS this year, in 2021. So congratulations for that.
Thank you.
And I will now let you take over and present your work. Thank you very much, Valentina.
Thank you. Thank you for inviting me to give this presentation today. And thank you for coming. So it's now almost 2 years that I'm not using a physical pointer. So this is, I don't know if I'm still able to use it. Okay, so as Rayad was saying in the introduction, I'm based at the Vision Institute in Paris, and my lab is an interdisciplinary group because we have both physicists and engineers here in blue, but also biologists and biophysicists here in red. And the interest of the lab is to develop and use new optical methods for all-optical manipulation of visual circuits. We work mainly in mouse brain. Okay, so before start to present you the results of my lab in the past year, I have prepared a few slides to introduce the idea of all-optical neuronal circuits investigation, which is probably not something that you are very familiar with. First of all, what is a neuronal circuit? So this is in a very large way of defining a neuronal circuit. Is a population of neurons that once activated can control a specific function. And a key part of neuroscience research today is to try to understand more and more on how those neuronal circuits work. And this could have many meanings. So we might want to know how different neurons in a circuit are connected. We might want to know which is the limited number of neurons that can control a specific behavior. We might want to know which are the mechanisms that a certain neuronal circuit use to send signal to other circuits or to process signal coming from other circuits. And so these kind of questions for years have been studied by using electrophysiological recording. So people use electrodes to both record these electrical signals propagating in the brain, but also to manipulate those signals. And the challenge today in neuroscience is to try to develop a number of approaches that enable to replace the use of electrodes with light, with the obvious advantage that this can be less invasive, and as you will see, also give access to the manipulation of thousands of neurons with the precision of the single cell. Okay, so the idea is to use light in both directions, so both for image the propagation of the electrical signal in the brain, but also to manipulate the propagation of these signals. And so this is also something that today in the modern language is defined as reading and writing the brain, or reading and playing the brain. Okay, so I will go relatively fast on the reading part, and most of the lecture will be dedicated on show you what we can do today using light to manipulate the propagation of signal in the brain. Okay, so from the reading side, so the question is, we want to use light to image to follow the propagation of the signal in the brain. So the signal I'm speaking now are these specific voltage signals that called action potential, which is the main signal that neurons use to communicate among them. And so if we want to use an optical microscope to visualize this electrical signal, the very first challenge, of course, is to be able to convert this electrical signal into a fluorescent signal that can be detected with an optical microscope. So this challenge has been nicely solved a few years ago with the development of what we called a calcium or voltage indicator. So here we show you the case of the calcium indicator. So the idea of those probes is that to make advantage of the fact that normally when you have an action potential propagating through neurons, this goes with the opening of calcium channels. And so during the propagation of this action potential, there is an important entrance of calcium in the interior of the cell. And so the idea of a calcium indicator is that people that developed calcium indicator was to find the way to engineer fluorescent probes that become brighter when they bind to calcium. And so if you can do so, each time you have an action potential propagating, you will see an increase in the fluorescent signal. So today we call genetic encoded calcium indicator because they can be expressed in a genetic way, meaning that you can very precisely control which kind of cell is going to express this specific calcium indicator. I will come to this point later on again. So here I will just show you an example. This is one of the very first movie that has been done by showing the potentiality of those probes. So here you see this is a neuron, and there is also an electrode to record the activity of the neuron here in red. So you see this spike that characterized by this just a spontaneous activity of the cell. And here in yellow, you see the corresponding fluorescent traces. So you see each time we have a trance of spike, we have a fluorescent signal. And this response is nonlinear, so when you have many of them, you can have as here, a very bright signal. Okay. So this was the first part. So we indeed can transform an electrical signal into a fluorescent signal, and so think of using an optical microscope to detect neuronal activity. Now, the next point was, can we detect the activity of a single neuron when this is embedded in a scattering tissue, as is the brain? And so for that, the second important discovery was the prediction many years ago from Maria Goppermeier of the two-photon effect, which probably most of you knows. But just in 2 words, the idea of two-photon absorption was that you can either an atom or molecules can be excited with the absorption of a photon, and that then give rise to emission of a photon as well. But you can also have the same process if instead of a single photon you excite your molecules or atom with 2 photons of roughly the half of the energy, and we will half of the energy and get the same absorption process. So in optical microscopy, there are 2 main consequences of this discovery is that because we use a lower energy photon, we can use a longer wavelength, and so this allows to penetrate more in scattering tissue because the scattering depends inversely on the wavelength. And the second important implication is that because we need this double process to happen, the absorption depends on the as you will see also later on again, on the photon density. And so if you quadratically on the photon density, and so if you realize a strongly focalized beam, eventually there will be only a very small region around the focal plane where the probability for two-photon absorption is high enough to produce a signal, and so this allows you to have an intrinsic optical confinement that we won't have in case of single photon excitation. And so the idea of two-photon microscopy was is now to use this small illumination volume and to scan this volume through a certain preparation and detect, for example, the calcium response. So for this, I have selected two examples because this is important to also for the rest of the lecture. So these are two examples in two cases where this two-photon scan imaging is combined with the use of this fluorescent calcium indicator. And this first film is taken in vivo from the visual cortex of a mouse, while different visual stimulation are projected are presented to the mouse, and you see that with scan imaging, with fast scan imaging, we can really record this activity. And so you will see there are cells that become brighter, there are group of cells, or they can be individual cells. And so each time we see something becoming brighter, it's because there is this electrical signal going through. And now there are, in two-photon microscopy, this is the basic two-photon microscopy when we do one plane at a time, but there have been a large number of laboratories that has worked to increase the speed, the volume, and the temporal spatial resolution to photon microscopy. For example, today, this is just to show you, we call about mesoscopy, so where we are able, as in this case, this is a spectacular film from the group of Svoboda at Jamila Farm, where they not only can record a specific region, but in this case, they can record activity from the entire visual cortex, again, here in mice. And then, of course, you can zoom in and decide the region from which you want precisely to detect activity. But we do have the capability to record from today thousands of neurons. And so this is really to make a very long story short. So today, on the reading side, I think we can really say that there have been a combination of many advanced optical methods. So we are on the way to record from millimeter cubic volume the activity of thousands of neurons with single spike precision and single cell resolution. Okay, once we have these fantastic tools, the next step was now that we can precisely see what happens in the brain during the propagation of signals, can we also use light to control the propagation of those signals? And so we are moving now into the writing part. So again, as it happens for reading, also writing activity using light, first of all, requires to make neuronal cell sensitive to light. And this challenge has been nicely solved by the revolution of optogenetics. And the idea of this approach is to use this genetically encoded microbial which are specific photosensitive channel or pump that can be expressed on the neuronal membranes. And once they bound to the retinal, the activation with visible light induce a conformational change on the complex, and this conformational change enable the entrance of positive or negative ions, and this in the physiology of neuronal cells means to what we say to depolarize or hyperpolarize the neuronal membrane, and this is exactly what happens in a physiological condition when the neurons are producing a spike. And so today, with light, we can, for example, here you see, if we repeat the photostimulation several times, so illumination with several times, we can not only produce a spike, but also control the spiking frequency. And if we have a propagating signal, we can switch off the light and inhibit the progression of the signal. So with the light, we have the capability to both activate and inhibit neuronal activity. So this is the opto part of the word optogenetics. And now why do we call this optogenetics? Because as was the case for the calcium indicator, those microbial opsins are inserted in a genetic way. So the idea is to build up, first of all, a genetic sequence where the specific opsin is bound to a specific promoter. Then this genetic sequence is inserted into the DNA of a virus. So in some cases, the virus can also be useful. And then they are injected into the mice brain. And now this virus, once entering the brain, they will infect the cells that they recognize on the base of the promoter that is in the sequence. So we can, by designing the sequence, define which kind of cell type is going to express this optogenetics protein. And so why this is important? Because now you have the capability to, for example, analyze the role that a specific cell type can have in controlling a specific behavior. So we show you to give you an idea of the powerful of this approach, I will show this. This is a very old film. It's one of the very first that was done at the beginning of optogenetics that started around 2005. And here the experiment was the following. They put these optogenetic protein in the motor cortex, which is the part of the brain responsible for the movement. And then they put an optical fiber shining visible light in the right part, which is controlling the movement toward the left. And now what you can see in this film, so this is the mice having a normal behavior, and then at a certain point you will see that there is some light going into the mice head, and the mice start to move. Okay, so with light, they could activate this specific population responsible to initiate the cascade that control the movement, and once the light is interrupted, the mice start again to have a normal behavior. And so you can imagine that the importance of this kind of approach today in neuroscience. And in fact, there have been a very large number of studies that's been done using a similar approach, with the idea to start really to map in a very precise way neuronal functioning with the precision of cell specificity that otherwise we won't have by using simply electrode. So in this case, of course, the impact of this technology is extremely important in neuroscience. But there is somehow a limitation in this kind of approach. So in most of the cases, an optogenetic experiment today is really done, as I show you in the film. So there is a big part which is to find the right promoter to target a specific cell type. And then in most of the cases, by using just an optical fiber shining visible light in large brain region, they get the precision in the manipulation because of the genetic targeting. But as you can imagine, if you do this approach, all the cells that are expressing a specific opsin are going to be synchronously activated, okay? Now, if you remember the example that I showed you before, for example, this is another example, still this is the visual cortex, you will see that even if all those cells are genetically identical cells, so they will eventually all express this specific opsin, but in a physiological situation, they never spike all synchronously, okay? But they have a very specific activity where they can have a very specific special temporal signature, and this is specific special temporal signature that contributes to the processing of the signal, and so to the control of the behavior. And so today, there is a large part of neuroscience where people are starting to ask questions, okay, given a certain cell population, is it possible to understand how those cells are connected among them? Is it possible, given a certain activity pattern, that we can record using calcium imaging to exactly reproduce the same pattern and see the impact that this is having on behavior? And so if you want to enter in this class of experiment, obviously you need something more precise than to simply shine visible light in a large region, but you want to have a manipulation approach that allows to get this precision in the produced activity pattern. Okay, and so this is we need then to transition from this sort of wide field illumination to what we termed a few years ago circuits optogenetics, meaning with this really a combination of approaches, as you will see in the following slides, that enable to control single or multiple neurons independently in space and time. Okay, so what do we need to reach this degree of precision? We need to, as we did in imaging, we need to be able to control the activity of a single cell deep in a scattering tissue. So the very first step was to replace visible light with two-photon excitation. The problem in optogenetics was that actually if you use a normal microscope, shining light in the small volume that I show you is used to do two-photon imaging, in most of the case, the number of channels that you can open is not enough to evoke an action potentials. And the second problem is that if you want to now control the activity of a specific group of cells using a scanning microscope, you won't have the temporal resolution to activate all those cells within a millisecond temporal frame, which is the time scale you would like to reach in biology. And so using two-photon excitation in optogenetics require to find solution to, from one side, increase the illumination spot because we want to open more channel instead than just shining a small spot, and plus find a way to illuminate multiple targets at the same time. And so for this, my lab years ago proposed to use two approaches that I show here. One is the approach of computer-generated holography, and the other is the general effect contrast method. I will mainly speak about holography because we won't have time to go through the both of them, but in the general idea with these two approaches is to use liquid crystal matrix to modify the propagation of the light so that at the sample plane, instead of being focalized just to a small spot, as it would be in a conventional microscope, we can actually control the shape of the light to precisely, to reproduce any arbitrary pattern. Okay, so in computer, I will now specifically speak about computer-generated holography, which is nothing more than a modern revision of an old principle that I guess you all know very well, which is the technique of holography that years ago was proposed and demonstrated by Dennis Gabor. And actually, the physics is the same, and so we can go through his drawing to understand how it works. And so here, the idea of this approach was to be able to reproduce with light the shape of an object in absence of the object. Okay, so how this can be? So the idea is to take a laser, to divide the laser in two beams, one that we call the reference beam, and another one that is illuminating the object that you want to reproduce with light. And now you get, at a certain position, you can record the interferogram from produced by the reference and the signal beam and the diffracted beam, and then by illuminating with the reference wave this photographic plate, this interferogram, you will create a virtual and a real image of the object. So the virtual image is the image that looking through the hologram will give you the perception of the object, but you can also put a camera somewhere in the real space and record the shape of the object reproduced by light. Okay, I'm pretty sure that you all know these techniques very well, and this was largely used, also gave rise to a big speculation in science fiction movie, where people start to dream about doing a sort of animated three-dimensional holograms where you can really build up the shape of an animated object. So today, with computer-generated holography, we are not so far from here, and the main difference has been to replace the physical object with an image template, so we don't really need to have the physical object. We can draw the shape that we want to reproduce by computer, and then you don't need anymore to really physically record the interferogram, but we can calculate with Fourier-based iterative algorithm the phase, the interference pattern that the light propagating from this object will create with your reference beam. And then this phase hologram is sent to the liquid crystal matrix, and if things are well done, when the light is going through the matrix, he's able to reproduce the shape that you have designed by computer. Okay, this approach was actually developed and demonstrated first by the people working in the optical tweezer community, but for us, the idea was to use this approach to precisely illuminate the neuron or the neurons that we want to activate. And so for that, we have to find a way to design this image template. And so for that, we propose to use the fluorescence, because in most of the case, when you target your neurons with a specific photosensitive channel, this goes also with a fluorescent reporter. And so as a first step, you can take a conventional two-photon image that this will show you where for example, this is you see your example here. So this is in case that we show the morphology of the cell. These are two different neurons, but also you can see, as we have seen before, the distribution of multiple neurons. And now on the basis of this image, you can decide, okay, now I want to selectively photostimulate this part or this dendrite or this group of cells. So you will physically crop the fluorescence image. You will take this part that is now the entrance of your iterative algorithm. This will give rise to a certain phase profile. And when the light is going through, as you will see, it's going to be focalized with a shape that exactly reproduces the shape that you want to activate. And because we are using a special lay modulator, you can also think of sending a series of holograms so that not only you can create a fixed shape, but you can also dynamically change your illumination pattern. And so you can imagine that now, because we have the capability to increase the spot size as we want, so this was a very nice approach for two-photon optogenetics, because we can think of now generating spots which are larger than this single spot, and so have a very efficient two-photon optogenetic, as we shown now back in 2010, that actually with this approach we could use two-photon in optogenetics. So something to be mentioned is that definitely by increasing the spot size, we increase the efficiency in the light stimulation. But for those of you who are familiar in microscopy, you will know that each time you increase the spot size, you quickly lose in axial resolution. And I will show you this here. So imagine now that this is an hologram producing on a sample plane a shape to illuminate a single neuron that typically is about 10 micrometers, or a shape that illuminates multiple neurons at the same time. And now if you look at how this beam propagates along the axial direction, as long as you want to activate just a single cell, the axial resolution is pretty good, so we have the capability for single cell stimulation. But if you want to excite a group of cells, you will quickly lose the axial resolution. And so we are going to lose the benefit of using two-photon microscopy. And so to perform multi-target illumination, we had to find a solution to keep the optical confinement even for large illumination shape. And so for that, what we propose is to combine with this approach the technique of temporal focusing, that probably some of you know. So this was is a very clever idea proposed back in 2005 independently by the group of Jaron Silberberg and Chris Zhu. And they proposed two-photon temporal focusing with the idea of performing wide field two-photon microscopy. So to understand the principle, we have to go back a little bit in key properties of two-photon absorption, that is the dependence of the two-photon absorption on the photon density. Okay, so in a conventional two-photon microscopy, because of this quadratic dependence on photon density, you can strongly focalize your beam, and so you will have the photon density is obviously much higher here than in the autofocus plane, and this is why you get optical confinement. Okay, this is why your signal will preferentially come from here. But we have seen that this small spot is not the best way to illuminate neurons expressing a specific optogenetic proteins, where we would like more to illuminate a large area, and so we won't have special focusing. And so the idea of temporal focusing was to still have an increase of the photon density at the focal plane with respect to the autofocus plane by playing on the temporal length of the laser pulse. Okay, so somehow we will see this after how this can happen. You can think of concentrating temporarily the photon at the focal plane more than in the autofocus plane, and this will also give you optical sectioning. So how can we do this? Actually, in practice, it's relatively simple, and all what you need is to put in your setup a dispersive grading, and so that the pulse of your laser is going to be diffracted by the grading. Because you have a pulsed laser, you have a certain number of colors around the central wavelength, and these colors are going to be diffracted through slightly different angles because they correspond to a different wavelength. And so now this will introduce a difference in the optical path among those different colors, and if you are familiar with the propagation of pulsed beam, this will obviously correspond to a progressive broadening of the pulse. And now if the grading is conjugated with the sample plane, this will give you exactly what we wanted, so a short pulse at the focal plane, while the pulse will become broader and broader and moving out of the focal plane, and so we will have optical sectioning for non-specially focalized beam. We can also see this in the temporal domain, and this is shown in this film, so you can forget about the color and now just think about your beam as made by different pulses. So because you arrive with a certain tilt, the intersection of your pulse with the grading will be, as you will see in this film, will be aligned. And so we will have, so this is the pulse propagating into the grading. So this is, if you would look at the surface of the grading, you will see a ultra-fast scanning line. And because this is conjugated with the sample plane, this is also what the sample plane will see. So actually it's as if we have realized an ultra-fast line scanning microscope. Okay? And so the axial resolution is the one of a line, and so this is why we gain in optical confinement. Okay, in our case, we wanted to also have holographic light shaping, and so before reaching the grating for temporal focusing, the light is going through a special light modulator, creating a specific holographic pattern, and so this is the propagation I showed you before, and this is what happens when you have the grating for temporal focusing, and you see that now you have nicely concentrated the absorption around the focal plane, and so you get axial confinement, this time independently on the lateral spot size. Okay, so if you want to activate one cell or 100 cells, the axial resolution will be always the one of few micrometers. Okay. Now, this actually to reach a true single cell resolution in optogenetics, the light shaping was not enough alone. It was also important to integrate progress from lab working on the engineering of opsin, and this I won't go too much into the details, but what was really important to reach a true single cell resolution was the recent development of what we call soma-restricted opsin. So now it's possible to not only control in which cell types the opsin is going, but also to restrain the expression only in the soma. And why this is an advantage, because now if this is combined with a precise light shaping, you're sure that only this cell is going to be excited and not all the surrounding cells that eventually sends a dendrite or an axon through the excitation volume. So I will show you an example of this, where this true single cell resolution was reached. This is now from my lab. Again, an image taken in vivo from the visual cortex of a mouse. You see that now we place 3 spots here, and by using calcium imaging, we record the photoevoked activity from these 3 cells, and then we can say, okay, now I want only that one get activated, and you see that actually only that cell is responding. So we are really on the way to control neuronal activity with a single cell resolution. So what's for? Do we really need to be so precise? And to convince you about that, I will show you an example. I will go relatively quickly on the biological story, but it's just to give you an idea of how far we can go with this kind of manipulation. So this is a project that we have in collaboration with a group of Rosa Cossart here in France, in Marseille. And her lab is really interested in improving the existence of what we call the hub neurons. And so this is back to an old theory from Donald Hebb that many years ago was predicting that actually the central function of the brain is not just a single neuron, but is represented by what we call the cell assembly. And so he was saying that actually there are specific group of cells that have a major role in coordinating the entire activity of a large circuit or in controlling function. And so if you can find those cells and you can control the activity of those cells, you might be able, with the control just one or few of those cells, to control the activity of a large network. And this is exactly one of the key questions of the lab of Rosa. Specifically, she's interested in demonstrating the presence of those cells in developing a brain. So we are now barrel cortex, again in mice. And the experiment was the following. They start by performing calcium imaging. So again, you see here conventional two-photon calcium imaging. You see this is a young circuit, so it's characterized by this super high frequency activity. And then the next step, on the base of this film, they could derive a sort of this functional connectivity analysis. So they were looking at how the activity of each of these cells is correlated with the activity of the other cells. And by doing that, they could find cells that are highly correlated with most of the cells of the rest of the circuit. And so those cells are those putative hub neurons, because they, no matter what they do, the entire circuits will follow. And so then the next question was, okay, if now I perturb the activity of these specific hub neurons, which is the effect of the entire circuits? And this is something you can see here. So we will build up in her lab an holographic system. And so this is the activity of the network, and at a certain point they start to place an holographic spot on one of those cells, which is expressing one of these opsin, and you see that the activity of the entire network strongly shut down, and this is also can be visualized here, where you have an important increase in the inter-event interval. So just again to cut a long story short, this was an important demonstration that indeed by acting just on the activity of a single neuron, there are situations where you can control the synchronicity of the entire network. Okay, so not all the circuits can be controlled just by acting on a single neuron. There are situations, and you will see example later on, where we might need to control more than a cell at a time, and eventually also cell distributed in 3D. So the next step from the technological point of view was to develop approaches that could deliver not only light on a single neuron, but to reach multiple targets eventually in 3D, but by keeping the precision of temporal focusing. And so for that, what we proposed was to now use two spatial layer modulators, so that you have, sorry, I went too fast. So to have a first one, as before, that is producing your holographic spot on the grating for temporal focusing. And then now we have a second spatial layer modulator that alone will produce only a distribution of multi-diffraction in the spot. And now when you have the two together, here you will have a convolution of this distribution with that one. And so basically the second spatial layer modulator will allow you to multiplex all what the first one is creating on the grating, and all of these shapes are going to be temporally focused. So I will show you an example here. So this is the case where we use this approach, in this case, to create 50 spots at 50 different axial locations. And because each of these is just the exact same replica of what the first SLM is creating on the first grating, they are all temporally focused. And so in the entire field of view, we had the axial resolution necessary to get single cell resolution. So the main message to say is that we can generate illumination shape to reach multiple targets, eventually in 3D, with a good axial confinement. And so again, is this really of use? And so for that, I select two examples, so a few examples. So the first two examples came from two other labs, not mine, but there are two very important papers that's been published in 2019. And where using holography, and here the question was the following. Again, in the line of this cell assembly, so can we found in the visual circuits distribution of cells that have a key role in controlling the visual perception, to the point that if I only activate those cells, I can create a visual perception. And so the experiment was the following. They start by training mice in having a behavioral task, because of course they cannot tell you what they see, so they have been trained in doing a certain task when they were stimulated by, when there was a specific visual stimuli that was consisted of grids of a perpendicular orientation. Okay, so the mice, when was seeing the grids in one way, was doing something, and the grids in another orientation were doing something different. Then the next step was to start to record, by doing calcium imaging, which kind of cells were activated during one visual stimulation or another. And then the third step was to remove the visual stimulation, or to decrease the contrast of the grids, and to go with holography in activating exactly the cell that were active during the visual stimulation. And interesting, in both cases, with few differences in the two approaches, they could demonstrate that the activation of a very reduced number of cells, down to 2, or anyway a very reduced number of cells, down to 2 to 30, it was possible to induce the mice in doing the correct behavior. And so the main conclusion was that actually this theory of this cell assembly is probably true. So there are probably circuits that we can really control by activating just a very reduced number of neurons. Okay, so I selected also another example now where we use the unique capability of holography to shape light in 3D. And this is when you want to investigate how signal propagate across multiple layers. But before entering into the example, I need to introduce another important field of application of optogenetics that I haven't mentioned yet. And this is the big promise of optogenetics to be used for visual restoration. So there are a very large number of retinal dystrophies which are characterized by a progressive degeneration of the photoreceptors. And so this, of course, leads to progressive loss of vision. And so the idea of optogenetics was to be able to design, to engineer a virus that can bring these photosensitive proteins in specific layers of the retina so that we can restore the conversion light to electrical signal and eventually restore the vision. And so now, okay, the retina is made by many, many layers. So if you are familiar with this, so of course the first layer are the photoreceptor, and where there is this first conversion light into an electrical signal, but then this electrical signal is sent to different cell layers, the bipolar cell layer, the amacrine layer, the ganglion cell layer. And so finally, after all this process, at the ganglion cell layer, there is the final signal which is created and then is sent to the rest of the brain for the right processing. So now the question is, where do we put these optogenetic proteins? Obviously, the most straightforward solution is to put it on the photoreceptor, because then all the rest of the circuit is intact, so your signal can be processed as in a physiological situation. But this is not always possible because there are situations where these photoreceptors are so deteriorated that they cannot express your protein. And so the next candidate is to go into the bipolar cell layer or even into the ganglion cell layer. So there are investigations in these three directions. And very recently, a few months ago, there's been an important paper published by joint collaboration by José Sael, director of my institute, and Boton Rosca in Switzerland, where they actually show that by putting an optogenetic protein in the ganglion cell layer, it's possible to restore basic vision in humans. Okay, so in this framework, you can understand that it's super important to understand how this signal propagate, because if you want to, for example, pattern the bipolar cell layer, you want to know which kind of pattern you need to propose to produce a certain output signal, and so on. And so in this framework, a few years ago, we started a collaboration with the group of Serge Picot at the Vision Institute, and exactly with this idea to understand how the retina processes different signals. They were interested in particular in starting by looking at a specific class of cell, a rod bipolar cell. These are cells which are implicated in the circuit responsible for the night vision. And the circuit is a highly divergent circuit, so morphologically high divergent circuit. And the question was, how local is the response? So if I activate one bipolar cell layer, what happens into the ganglion cell layer? So how sparse is the signal, or is this local? And so for that, the first step was to develop a promoter, so that, as you can see here, we can send the optogenetic protein selectively only in this kind of cell. And then the next step was to build up an optical system where we could activate a different bipolar cell layer at different location while looking with calcium imaging at the evoked response into the ganglion cell layer. And so for that, using holography was of key importance because those two layers are separated by 50, 70 micrometers. And so we need to decouple the imaging from the photostimulation plane. Okay, so I will go through quickly through the experiment. So this is, you see, an example. So the idea was then to start perform calcium imaging here, so the ganglion cell layer, look at how those cells respond, while different patterns of holographic photostimulation are presented down into the bipolar cell layer. We did this for many mice, many field of view, and at the end, the story was to analyze all this data and to find how local is the propagation of the signal. And the interesting results was that actually, given a certain ganglion cell layer, we can control the ganglion cell, we can control the activity of this cell only if the bipolar cell excited is within a very reduced area, about 1,500 micrometers. And so this enabled to conclude that actually this circuit, despite its divergency, has a very limited spatial divergence. And this is interesting because if you want to use this approach to know which kind of pattern to reproduce in the ganglion cell layer, we know what to do in the bipolar cell layer. Okay, so this is another example where the capability of shaping light in 3D was important. And now, before to conclude, I want just to move in the last topic that I haven't mentioned yet. I will go quickly on that, but this is the how can we integrate in this neuronal circuit manipulation also the temporal resolution, the time code. Okay, to introduce this idea, I will show you an experiment first. Again, this is an experiment using holography, unfortunately not from my lab, it's New York University. And this was a very important paper because here they did, again, very similar to what I showed you before. here the question was, now we are in the olfactory bulb, and the question was how many cells need to be activated to give, again, in this case, an olfactory perception, okay? So again, the mice have been trained in recognize a certain stimuli, and then in performing a certain task, if they receive or not, if they feel or not this stimuli. They use, again, holography. And what they did was to start first to send a large illumination spot, and then to reduce them more and more the number of neurons that they are activating. So again, the importance of single cell resolution. And doing this, they could show that actually the mice was still doing a right response when they activated between 20 and 30 neurons. So again, these important results that only the activity of few neurons is enough to give a certain perception. So then they did something more, and so they start to ask another important question, and this is, okay, once I decide that this specific cell assembly, these 20, 30 neurons alone can give you the right perception, now the next question is how synchronously they have to be activated to give you the right perception. So they have to be really synchronized, or we can slightly detune in time the spiking and still have the perception. And so for doing that, they compare the situation where these 20 neurons were all activated at the same time. We did 10 milliseconds. And then they, instead of having just one hologram exciting all the neurons, they decompose the group in 2 or 3 holograms, and then they send those holograms with us in a certain temporal sequence. And what they show that just the desynchronize those stimuli with 30 or even 50 milliseconds was enough to completely decrease the accuracy of the response. And so this again goes together with the famous Donald-Hube prediction that neurons that fire together wire together, but the question is how short is this together, okay? And so here it seems that it has to be at least shorter than 10 milliseconds. And so, but if you want to go more into the details of that, the ideal experiment we would like to have at a certain point is to be able to record in a very precise way the activity of a specific circuit, and then to play back exactly the same activity, and then to start to say, if instead to play back exactly the same activity, I start to slightly desynchronize the stimuli of 1, 2, 3, 10 milliseconds, and see if this is enough to change the behavioral response. And so what is the technological limit to that today is the resolution we have for fast sequential patterning. And this resolution is due to the fact that, as I was telling you before, to create this sequence of a pattern, we need to project multiple holograms. And there is a limited temporal resolution for that, which is fixed by the refresh rate of the liquid crystal. So that today, this is between 60 and 500 hertz, and which correspond to 1 to 20 milliseconds. And on top of that, there is also the cell illumination time. And so if we want to go faster than that, we need to find solution to speed up the resolution for sequential patterning. And this is the last setup I will show you. And this is the last variant we propose for homolography. So it's the same system as before. So we have the grating for temporal focusing, the special ray modulator for this multi-target multiplexing. But now what we did was to insert a galvanometric mirror in the system, and then to address the special ray modulator instead to use just a single hologram producing your multi-spot, to address the special ray modulator with a number of holograms. And now you can imagine that each of these holograms is, for example, producing a spot or a group of spots. And now what you can do is to scan your temporally focused line across those holograms. And then in this case, you can go at the speed only limited by the galvanometric speed. And so by doing that, we can actually generate this very fast sequential light patterning. Okay, to really show you all what we can do with this stuff, we would need another half an hour probably, but I just wanted to show you that actually also the time is an important parameter, and the technology is arriving to that now. because also what was missing is not only the optics, but also opsin that could respond in a very short illumination time, and this also has been an important revolution. It's something which is happening now in the last 5-6 months. So we have opsin that can produce an action potential within less than a millisecond. Okay, so this is now time to conclude, I guess. It's a general conclusion. The general conclusion is there have been really multiple disciplines together that have contributed today in giving us the technology for reading and writing neuronal circuits, and to do this in a very precise way. So the number today that we can claim is that we can control large number of hundreds of neurons in millimeter cubic volume at millisecond temporal resolution and single cell resolution. And so this is really opening a number of new exciting questions in neuroscience. And then there is another conclusion that I would like really to mention, because this is, for me, is an important point, and this is the interdisciplinarity in the research. So all this story, as you see, has been written by people coming from completely different fields, people that has developed the opsin, people that doing photonics, and people that have the right question in neuroscience, and the biophysics, and the people with expert in genetics. Laser development was also an important point. And so this is something that I really care to mention each time, the importance of interdisciplinary research. So I want to thank again all the people from my lab. In red are the ones that specifically contribute to the data I show here, but all of them are contributed to many exciting research we are doing. I already mentioned the different collaborators and the financing source. And obviously, thank you for your attention.